Hello grade 11 students. In this video we're going to talk about our new lesson, Spermatogenesis. This lesson is document 4 for chapter 1, the basic mechanism of sexual reproduction, document 4, Spermatogenesis. So first, what is spermatogenesis? Spermatogenesis is the process by which the mother cell of the gametes diploid 2N gives rise to the haploid 1N male gamete, which is called the sperm cells. It takes place inside the testes, starting from puberty until Leydig cells. Leydig cells, they are somatic cells, 2N, 1Q. They are located among the seminiferous tubules. Already we discussed this in the video before. And the major role is the secretion of the primary sexual hormone, the testosterone. So the Leydig cells, they are somatic cells, 2N, 1Q amount of DNA. They are located among the seminiferous tubules. They have an important role in the secretion of the primary male sexual hormone, which is called the testosterone. Sertoli cells, also they are somatic, 2N amount of DNA is 1Q. They are located among the seminiferous tubules. They are located inside, the seminiferous tubules among the reproductive cells. They support, they have the role of nourishment, and they sustain the reproductive cells. Mother cell of gametes is 2N, 1Q. It's called Spermatogonia. They are located close to the basal lamina of the seminiferous tubules, and they give rise to the haploid gametes. So the mother cell of gametes, it's 2N, 1Q. Mother cell of gametes, it's called Spermatogonia. They are located close to the basal lamina of the seminiferous tubules, and they give rise to the haploid gametes. First, let's move to the stages of the spermatogenesis process. We have different stages. Let's start with the first stage, multiplication phase. Multiplication phase is the phase where the number of spermatogonia is amplified, increased in number, by the process of mitosis to produce many spermatogonia, each having 2N chromosomes. Each is made up of 1 chromatid, so they are 2N, 1Q. Second phase in the spermatogenesis process is called the growth phase. Before we move, let's move to multiplication. Let's repeat it. It's the phase where the number of the spermatogonia is amplified and they produce several spermatogonia, each having 2N chromosomes, and it's made up of 1 chromatid, so the amount of DNA is reduced into half. Let's move to growth phase. Growth is the interphase. It's the phase of the cellular development. It's the alarm. So spermatogonia, they give rise to larger cells. These cells, they are called spermatocyte 1. Spermatocyte 1, they result in the growth phase, in the interphase. They are called also primary spermatocytes or spermatocyte 1. They are 2N, 2Q, and 2 chromatids per chromosome. So as I said, in the growth phase, the development of the spermatogonia will occur and they give rise to larger cells. They are called spermatocyte 1 or primary spermatocyte, 2N, 2Q, and the chromatids, they are 2 chromatids per chromosome. Let's move now to the maturation phase. Maturation phase, or it's called the meiosis. Meiosis, as you know, we have meiosis 1 and we have meiosis 2. That's why we explained the meiosis in the video before. By the end of meiosis 1, spermatocyte 1 will produce 2 spermatocyte 2 cells. They are called secondary spermatocytes. They are 1N, 1Q, and 2 chromatids per chromosome. While by the end of meiosis 2, spermatocyte 2 will produce a new type. It's called spermatid. Spermatid, it's one N, the amount is Q/2, and one chromatid per chromosome. So by the end of the maturation, we will have 4 spermatids. Since we have 2 spermatocyte 2 are produced after meiosis 1. After meiosis 2, every spermatocyte 2 will give 2 spermatids. So we have 2 spermatocyte 2, so we will have 4 spermatids. Okay? Maturation phase. Maturation, by the end of meiosis 1, we have spermatocyte 1 produces 2 spermatocyte 2 cells. They are called secondary spermatocytes. As I said, 1 N, 1 Q, 2 chromatids, chromosome. While in the differentiation phase, which is the last phase, is the phase where the spermatids, they will give rise to mature and functional sperm cells. So this is the phase where we'll have functional and mature sperm cells, 1 N, Q/2, and 1 chromatid per chromosome. So the produced sperm will be released in the lumen of the seminiferous tubules. So this is the figure. This is the acrosome. It's called the acrosome. It's full of enzymes. This is called the acrosome. So this result from the Golgi apparatus. This acrosome is a structure which is rich in enzymes and it covers the nucleus. We have distal centriole give rise to the flagellum, to the tail, to the flagellum of the sperm. We have the mitochondria will group in helix in a loop, as shown here, spiral shape, and will group in a helix around the sperm, around the flagellum, exactly, and will form the midpiece or the middle piece. And most of the cytoplasm is going to be released outside as something called residual body. So as a revision for what we have explained, the spermatogenesis includes the multiplication phase, is the phase where the number of spermatogonia is amplified by the process of mitosis. This will lead to a different number, to a high number of spermatogonia, each having 2 N chromosomes, each made of 1 chromatid, so 2 N, 1 Q. Growth phase, as I said, its interphase is the phase of the cellular development, enlargement, so that the spermatogonia will give rise to larger cells. They are called spermatocytes 1, or we can call them primary spermatocytes, 2N, 2Q, 2 chromatid per chromosome. And in the maturation phase, it's meiosis 1 and meiosis 2. After meiosis 1, it's done. We have spermatocyte 1 will give rise to 2 spermatocyte 2. It's called secondary spermatocytes. They are 1N, 1Q, 2 chromatids per chromosome. While after the end of meiosis 2, every spermatocyte 2 will give rise to 2 spermatids. So we have 2 spermatocyte 2. They will give rise to 4 spermatids. These spermatids, they are 1N, Q/2. It's divided over 2. And 1 chromatid per chromosome. We have 4 spermatids at the end. And the last phase is called differentiation phase, or it's called spermatogenesis. It's the phase where the spermatids will give rise to sperm cells that are functional and mature, 1N, Q/2, and 1 chromatid per chromosome. The produced sperms will be released into the lumen of the seminiferous tubule. As I said, during the differentiation phase, the spermatid undergo several changes. It's called differentiation or spermatogenesis. The Golgi apparatus, as I said, it gives rise to the acrosome, the structure rich in enzymes and covers the nucleus. The distal centriole give rise for the flagellum, it's like the tail for the sperm in order to allow them to move. And the mitochondria will group in a helix form and around the flagellum and form the midpiece or the middle piece, and most of the cytoplasm is going to be eliminated in the form of residual. So as you know, Spermatogonium, it's 2n equal 46, number of chromatids, 1 chromatid per chromosome. Spermatocyte 1, 2n equal 46, number of chromatids, they are 2 chromatids per chromosome. We have also the spermatocyte 2, 1n equal 23, it's 2 chromatids per chromosome. Spermatid 1n equal 23, 1 chromatid per chromosome. And the sperm cell 1n equal 23, it's 1 chromatid per the chromosome. Aneuploidy. What is the aneuploidy? Aneuploidy is the presence of abnormal number of chromosomes in a cell, like in the case of we have an extra chromosome or a less chromosome number. For example, in a cell having 45 chromosomes and less in 1 chromosome, or 47 instead of having 46. So this is called aneuploidy. Azoospermia. What is the azoospermia? Azoospermia is a medical condition of a man whose semen contains no sperm. It's mainly associated with infertility, but many forms are amenable to medical treatments. In humans, azoospermia affects about 1% of the male population and may be seen in up to 20% of male infertility situations. So this is the normal case. We have high number of sperms in the normal case. While in the azoospermia, we have either no or very low number of sperm cells shown here. So this is the sperm count. Here there is no sperm found in the semen. Oligospermia, oligozoospermia, and low sperm count. All of them refer to the semen with low concentration of sperm and is a common finding in male cases of infertility. Often, semen with decreased sperm concentration may also show significant abnormalities in the morphology of the sperm or in the motility. Technically, it's called oligozoospermia. Okay, oligozoospermia. So, abnormalities in the sperm morphology, shape, and the motility. So, I repeat, oligozoospermia, and the low sperm count refer to the semen with low concentration of sperm and is common finding in male infertility. Often, semen with decreased sperm concentration may also show significant abnormalities, either in the sperm morphology or in the motility. Technically, it's called oligozoospermia. There has been interest in replacing the descriptive terms used in semen analysis with more quantitative information. So here, the abnormalities in the morphology or in the motility sometimes. So this is the normal head. Here we have a large head as seen here. This is a very small head. This is 2 heads sperm. And here we have 2 tails. As you see here, we have, this is the first tail and this is the second tail. So 2 tails sperm. Here tapered head, as you see here. And here we have abnormal midpiece. It's very thick here. So these are abnormalities in the morphology of the sperm and may affect the motility of the sperm cells. So you have to know these conditions. You have to know that aneuploidy is the presence of abnormal number of the chromosomes in a cell. For example, having 45 or 47 instead of having 46. You have to know that azoospermia, where we have no sperm or very low number of sperm in the semen of the male, and it affects the male population in about 1% and may lead to 20% of the infertility situations of males worldwide. And you have to know that the oligospermia or oligozoospermia and low sperm count may refer to the low concentration of sperms in the semen, and sometimes we have abnormalities at the level of the morphology or at the level of the motility, mainly referred to as oligospermia, and this is the cases shown in this figure. So you have to study well the notes regarding the steps of the spermatogenesis, and you have to know what happens in every single stage, in addition to the information related to the amount of DNA, diploid, haploid, and so on. And you have to know that the Golgi apparatus gives rise to the acrosome, which is a structure which is rich in enzymes and covers the nucleus. You have to know that the distal centriole gives rise to the flagellum of the sperm cell. The mitochondria will group in a helix around the flagellum and form the middle piece or the mid piece of the sperm cell. And you have to know that most of the cytoplasm is going to be eliminated away in the form of residual body. Also, you have to know that the spermatogonium, it's 2 unequal 46 in the number of chromosomes. Number of chromatid per chromosome is 1. Spermatocyte 1, 2 unequal 46. You have to know that we have 2 chromatids per chromosome. Spermatocyte 2, you have to know that 1 n equal 23. 2 chromatids per chromosome. Spermatid and the sperm cell, they are 1 n equal 23 and 1 chromatid per chromosome. Okay, thank you. Bye bye.